So, first of all, I like to know what inspired your team to explore terbium and bile salt gels for enzyme detection, and how did this idea evolve over a decade?
Okay. Okay. This is actually a long story. I don't want to take a lot of time, but I need to tell you a little bit, about this. It turns out that these lanthanides are used in many display devices. For example, probably on the screen that you and I are looking at, mobile phone screens, mobile phone electronics, they have a lot of lanthanides. And of course, as you know, that these days we are also talking about problem of not getting lanthanide-derived magnets. So lanthanides are very important. But it turns out that lanthanides, while they have such properties, they don't show this photoluminescence in water. For example, imagine this is a lanthanide ion. And if it is present in water, it will not show any emission. So that is basically the luminescence or photoluminescence or fluorescence. So over the years, people have figured out that the way to do it is to get rid of the water by surrounding it with something. Okay, what is called in chemical terms a ligand. But that's not enough. You are only removing the water. Now what they do is they add an antenna. So this antenna absorbs the light, transfers the energy to the lanthanide, and now the lanthanide is happy and gives out light. Okay, so this is a principle that was known. Now, we found out many years ago that, in fact, this is some work that was published in 1914 or 1913, that a very common surfactant called biosurfactant, called bile salts in general, these are molecules which look like this, sort of a bird's beak, and one face is non-polar or hydrophobic. The other face is polar. Now, this is the amphiphile. Amphiphile is like a soap or a detergent. But the difference is that here one face and the other face, they have different polarities. So these are called facially amphiphilic molecules. Okay. Now, why is it important? It is actually formed from cholesterol in our liver and accumulate in the gallbladder. And when we eat things with fat, for example, now fat is not soluble in water. So to metabolize fat, to assimilate fat in the body, we need to make an emulsion. Just like, we use a cold cream or different kinds of body lotions, where it's basically an emulsion of a fat or an oily material with water. So this emulsification of the fat that we eat is done by the bile salts, in part. So this gentleman from UK, he found out many years ago, a century ago, that when you mix these bile salts with calcium, it forms a gel-like material. Gels are very important in our everyday, usage of different types, in cosmetics, in medicines, in food, for example. Okay. Kids love jellies and jello and things like that. So our interest was that can we make this gel instead of calcium with other metal ions? And this was initiated, in fact, in 2010. So what we found quickly, again, I will summarize, that many metal ions other than calcium also form these gels. So what happens is that, for example, ions like copper, cobalt, cadmium, and eventually we found that all the lanthanides which are commercially available, they also form these gels. Okay. And that then made us think that, fine, now the gel is in a, let me put it this way, the ions are embedded in the gel. And instead of this one, the so-called ligand, if I now bring in just an antenna next to it without physically connecting them, but they are held together in the gel, will it have the same effect? In other words, will it now absorb light and transfer it to the lanthanide, and the lanthanide will emit.
Mhm.
And very quickly, we found out that this, in fact, can be done. So that opened up a lot of possibilities, that using these colorful gels, we could tune the color depending upon the nature of the ion. For example, we can use terbium for green light. We can use europium for red light. And we can have a mixture of them, and we can tune the color from all the way from green to pink to yellow to red.
Okay.
And then the idea is that, okay, fine. So now we have these two, and this can transfer, makes the gel colorful under light, under excitation, under UV lamp. Now the question is, what do we do with them? So our idea was that, okay, I will somehow block the antenna so that it cannot transfer the energy. I'll block it in such a way that it can be unblocked by a specific trigger. And the specific trigger can be, let me go out and zoom out a little bit. So the specific trigger can be either a small molecule, such as hydrogen peroxide, or it can be an enzyme. So if I block it in a certain way, that can be removed only by a specific enzyme, then basically I have a way of detecting the enzyme, because enzyme unblocks this, releases the antenna, and then the antenna can trigger luminescence. And now my lanthanide is happy. So this is the basic principle that we have used. For different enzymes, for different small molecules like hydrogen peroxide, formaldehyde, fluoride, we just change the way we block it, so that the unblocking is very specific to the enzyme or small molecule, which basically act as a trigger. Okay. So again, if I block the antenna, it cannot absorb light and transfer to the lanthanide. But when I unblock, then it transfers the light, and my lanthanide is happy and emitting in the green if it is terbium, and red if it is europium.
No, but
That's the
the
principle.
initial idea, from where this idea originated to use this such things and for enzyme detection?
Okay, this was in fact, well, as soon as we discovered that these sort of antennas can trigger photoluminescence, we made these gels, which were very colorful. We made a lot of studies with them in terms of making nanoparticles for certain kinds of other types of applications. But then we thought that we should look for something that can benefit the common man in the long. And again, this idea came, just like that. Sometimes, because what we thought is that this antenna has some appendages, okay, through which it can interact with the lanthanide. These are called ligating groups. So the initial idea was very simple, that can we modify them with an enzyme cleavable group? Well, I had some early interest in enzymes, enzyme inhibitors. This was part of my postdoctoral work long ago. So I have been familiar with at least the simpler enzymes. We have not done any work on enzymes subsequently. So I was familiar with certain kinds of enzymes which can do simple hydrolysis reactions. This is something we also teach, because enzymes and proteins are also one of the 4 types of macromolecules of life. So that's how the idea came, that we can block the appendages and see whether it can be unblocked by a specific enzyme. So the first thing we tried was for an enzyme called lipase. That is also an enzyme which is important in our body. That was in 2012. And we showed that indeed this principle works. So similar to this, we then followed it up with many other enzymes, including some which are important biomarkers.
Right.
So I hope I have answered.
Yes, perfectly fine. So if you don't mind, can you please come closer to the mic? Your voice is very dim.
Oh, is that right? Okay. In that case, I would probably try to
Raise the volume level if it's possible.
Yeah, it seems to be Seems to be okay. Let me just check the audio level, microphone input level. Okay, I'm increasing it. It seems to be maximum. Automatically adjust microphone volume. Yeah, that is also set. What I will do, I'll very quickly then zoom out a bit. Yeah, so that I can come a little closer and my head doesn't look too big. Okay, is it better?
The same.
Same?
Yeah.
Uh,
I don't know.
Yeah, I don't have any other microphone, but I'll try to come a little closer.
Closer, better.
Yeah.
Okay. Can you elaborate what is 2,3-DHN and why it was chosen as the antenna molecule and how its chemistry complement terbium ions?
Okay. See, what happens is I mentioned about this antenna. I mentioned about the metal. Now, in order for this to absorb energy and transfer it to the lanthanide, the metal ion, it must have an energy higher than the lanthanide's energy.
Okay.
In other words, I can transfer energy only from here to here. Okay, because this has to be higher. Suppose this is the antenna, this is my lanthanide. So I need to have an energy which is higher than this. Okay, because if it is lower, it doesn't have enough energy to transfer, right? So I need to have their energy levels matched in such a way that my donor, the 2,3-dihydroxynaphthalene, is basically a donor because that absorbs energy and passes it on to the acceptor. This is my acceptor. So that's how 2,3-dihydroxynaphthalene, it turns out, we had it in our lab for some other purpose. And based on earlier studies by people who had used naphthalene and naphthalene-like molecules, we chose 2,3-dihydroxynaphthalene, only this specific isomer, and it seemed to work. So I think we were pretty lucky in getting the first attempt to work with the terbium-based system.
Okay, got it. And how does the gel matrix derived from bile salts enhance the performance of the sensor, particularly in terms of signal clarity and stability?
Okay, so this is again a slightly involved, slightly longish answer for this.
Okay.
Because when the gel forms, what happens is we use about 1% by weight of the material which forms the gel. And the gels form a sort of a network of fibers, sort of like a ball of cotton. It's sort of like an entangled threads, something like this. I don't have anything else to show you. So these sort of fibers are formed in any type of gel. For example, if I take let's say a shower gel and look at the electron microscope, I will again see entangled fibers there. So these fibers are formed by our biodetergent, that is a bile salt, and the metal ion. Because the bile salt has, again, a tether, what is called a carboxylic acid at the end. So that connects to the metal ion and leads to fiber formation. Now, when we add the antenna doesn't stay in solution. Antenna sticks to the fibers. Okay, so that brings the antenna and the lanthanide in close proximity. And this is our working hypothesis that when we make the gel, it is basically a self-assembly of three components: the bile acid or bile salt, the metal ion, and the antenna. So they assemble together, form this kind of a fibrous network. Okay. And so on the fiber, I have a metal, the bile salt, and the antenna. So they come close to each other. And that assists in this energy transfer and the resulting photoluminescence.
Okay. Were there any significant hurdles in designing the paper-based format, especially in maintaining the structural integrity and fluorescence efficiency?
Yes, this is something that we had to do by trial and error. In order to simplify the testing, we thought the paper-based sensing is probably going to be easier, less expensive, and more easy to handle. Yes, the gel, when we make let's say in a test tube, that gel it's largely water and about 1% of the solid material, which has formed like a fiber. But when we coat it on paper, the water disappears, and it forms what is called a dried gel, which is called a zero gel. Zero spelled with X. Zero gel. So we were initially not very sure whether the zero gel will retain the property of the wet gel and will also retain the reactivity, will retain the photoluminescence. But again, we were lucky. With some trial and error, we also looked at these coated gels. So we take a paper piece, a small piece of paper, basically cut from a filter paper using the common office punch, which is used for making holes in the paper. So we use that as a very low-tech way to make small paper discs. And we had about between 20 and 30 microliters of the gel and coat it, dry it, and then use it directly. And this coating we have looked at by microscopy, and we find that it retains a fibrous structure. And what happens is that the paper also has cellulose fibers. So gel fibers and the cellulose fibers of the paper, they get integrated. And so the paper can hold the dry gel. Sometimes we have to make sure that there is enough moisture in the coated gel in order to have the in order to retain the effectiveness. But by and large, we have been reasonably successful in maintaining, and in a few cases, we see, in fact, a better sensitivity with a paper system than with the gel system. And that varies on a case-to-case basis. I'm sorry.
So how user-friendly is the procedure for non-experts?
It depends upon the particular protocol we follow. Again, the simplest protocol is everything is on the paper. Everything is coated on the paper, and we simply add the analyte, which is basically, let's say, a biological sample. Can be blood serum, can be urine sample, it can be anything else. Wait for some time, and then look at its photoluminescence. So that is the simplest one. But there are a few cases where the reaction on the paper is slow. So in that case, it involves a slightly involved protocol in which we pre-mix everything and then make the gel and coat the paper. That requires a little bit of expertise, but then that's not very difficult. It's like mixing A with B and wait for 5 minutes and put it on the paper and then measure it. It will require a little bit of a skill, but it doesn't require a very big knowledge of how to do it or what is happening and all that. So the protocol is very simple, but it involves a small, maybe a 2-3 minutes of a prep, like many other methods, because ultimately, whenever you take a biological sample, let's say throat swab or a blood serum or urine sample, there will be a procedure before you can actually see the measurement.
Right.
That way, I believe that this is, there is still scope of improvement, but as of now, it looks good that this can be, very user-friendly technique.
For non-experts as well. Okay, have you begun or planned any clinical collaborations to validate the sensor with the patient samples?
Okay, that's a very good question. In fact, very soon after the news came out in the paper, we have been contacted by at least 2, maybe more, 3 persons. Some of them are interested in taking it forward for clinical validation. As of now, we only had preliminary discussion, but it is absolutely important to have a clinical validation to make sure it works on real life samples.
True.
And it can give a readout which can compete with existing methods. But we are optimistic that once we do the clinical validation with different types of samples, we can have something that is practical, useful, and inexpensive.
So, given that gel, this glucuronidase is also linked to other cancers and conditions, so how adaptable your sensor for broader clinical diagnostics beyond liver cancer?
Okay, let me go back one step. This particular enzyme, beta-glucuronidase, is an important enzyme because what it does is that a lot of harmful substances that we get in our body, some which naturally come to us, for example, the pollutants, and certain other things like in the form of drugs, in the form of other chemicals, which are a lot of foreign to our body. These are collectively called xenobiotics. That connection? I think we lost the connection for a few seconds.
Okay. Xenobias you mentioned last.
Sorry?
Xenobias.
Xenobiotics. So these are foreign substances present in our body, and the body must get rid of them. That also includes medicines, for example, paracetamol that I have taken. At some point, the unused part of it has to be removed from the body. But many of these drugs, other xenobiotics, toxic materials from, let's say, pollutants, these are not soluble in water. So that's where attaching a soluble or water solubilizing group is very important. And this is what is done by another enzyme, which attaches a glucuronic acid, a sugar-derived acid, to the xenobiotic drug or toxins, so that it can get into our intestine and get eliminated either with the stool or with the urine. But if we have a high amount of beta-glucuronidase, then it cuts the solubilizing group and makes the toxin back into its toxic water-insoluble form. And that is what sometimes causes the toxicity. Now, beta-glucuronidase, the elevated level of beta-glucuronidase, it is not only observed in liver cancer, but in other types of, for example, breast cancer, colon cancer, and so on. And I think prostate cancer as well. But I must say that cancer is a very complex disease, and one should not rely only on one biomarker. But based on some of the recent findings that we have seen in the literature, beta-glucuronidase is an important biomarker, but for several types of cancer. So an early detection might signal that there is a problem. But of course, eventually more studies, more analysis, perhaps in some cases biopsies are needed to sort of confirm. But testing a biomarker, particularly when it can be done in a non-invasive manner, taking a blood sample or a stool sample, that can give an early indication. And that's basically what we are looking for with this simple method. But again, I must say that a clinical validation is super important.
Right. So how does your sensor's limit of detection compare to existing commercial liver cancer diagnostics in terms of sensitivity and reliability?
Okay, this is again another important question. We have not done a very detailed comparison of this because we felt that this is to be done when we start the clinical validation. We have not, in fact, looked at the existing diagnostic labs in detail because we kind of postponed that for this purpose. But I should also mention that our intention is not to create a world record for detecting the lowest amount of beta-glucuronidase, but we would like to make sure that it can detect a low or a high level found in any biological fluid. And I think our detection level that we reported compares reasonably well with some of the already published methods.
Okay.
And I should also point out that our method has the advantage because of this lanthanide. The lanthanide has a long lifetime for its emission. Let me try to give an example. Let's say I have two light bulbs, okay, which has a red light, one which gives a, let's say, yellow light, and I would be looking at only the yellow light, okay. So if both lights are lit at the same time, if both are glowing at the same time, then my eye will not be able to focus exclusively on the one that I want. Okay. So this way, so if I'm using any biological sample, there may be some other components which might give a light of a different color, and that might make my vision a little bit confusing because I want to look at this, but there's some other stray light coming here. So the lanthanides have the advantage that, let's say, this light glows for only 2 seconds. Okay, most of the biological lights, I mean fluorescent molecules, they have a very short lifetime. So imagine that this will go for 2 seconds and this will go for 20 seconds. Now but if I start looking at, if I keep my eyes closed and open my eye after 2 seconds, the red light will be gone. Okay, because both, so as soon as I shine under UV lamp, I see both the lights, but I open my eye after 2 seconds. And by that time, the red light is gone. And therefore, I can see this one much more accurately, and my eyes will be much more sensitive to the light I'm seeing. So this is also called gated measurement. So I open my eyes after this fellow has stopped emitting. Okay. That's where the lanthanides have a big advantage. So that's something that we feel that has an edge over even other fluorescence methods because of the sensitivity. We can optimize things a little bit more to make it more sensitive. We are actually developing a small equipment, specifically, which is a portable equipment. We already have a portable equipment, which we have used for the paper-based sensor. We are trying to develop a second generation, which will have higher sensitivity, lower sample volumes, and so on. But we are not there yet.
Right. And in the end, want to ask, what would be the next major milestone or optimization required before this sensor can be commercialized or made widely available?
Okay. Two things. One is that, as I mentioned, we have a specific blocker of the antenna. So that blocker is designed by us. That is our sort of like our USP here. Okay. So, and this blocker, we have to find a way of synthesizing it or making it in a much more efficient manner. Because in the first phase, our interest was only to get to this. Okay, we didn't bother about the efficiency. So if at some point this becomes a really viable method for detecting enzyme in a clinical setup, then we have to make it more efficiently so that the cost comes down. We don't have any cost analysis because we need very small quantities of this. But it's also made from very easily available materials, so that we believe the cost may not be very high, but we still need to optimize this. So that is one aspect. Second is to improve the sensitivity and develop perhaps a more sensitive portable equipment, and that can be done. That we have already shown that it can be done because normally for fluorescence measurements, you use what is called a plate reader. In clinical labs, and biology labs, there is a plate reader. We don't need, and that costs quite a bit of money, 15 to 20 lakhs. Whereas the prototype we made was probably 1 lakh or less. And but then we are trying to optimize it so that it can give more information at a lower price. And I think that is possible. But last but not the least, the most important thing is the clinical validation. because the method has to work on real life samples.
True. Right, sir. Thank you so much. Thanks for your insightful views, and you described everything so nicely with the examples. Thank you so much. Thanks a lot for your
Thank you
time,
very
sir.
much. Thank you. It was a pleasure talking to you.
Same.
Okay.